The Variant Analysis app can be accessed from the Zetabase landing page after logging in. From here, we can explore all samples within each study through the sample browser. The active study can be selected from the Population Study drop-down menu here. And in the main table, we can see all individuals, files both loaded and generated, and analysis cases associated with each sample. In a similar manner to all IVA browsers, these can be filtered using the options on the left-hand side. And more information about the selected sample, which is highlighted in green, can be seen under the main table. For the purpose of this video, I am going to use the preloaded 1000 Genomes project samples. Here in the main table, you can see we have 3032 records. We can access the variant browser from the main menu bar up here, or from here, which will return us to the main Variant Analysis app landing page. In the variant browser, you can explore your dataset in the context of all variants held within Cellbase for your project. Here you can see we have over 138 million records. Variants can be filtered using the filter panel on the left-hand side, using a range of options. We can filter by frequency within your active study, here are 1000 genomes, and we can select individual subsets of this or the whole population, or other studies within the same project. We can filter by genomic context, such as using chromosomal coordinates, which could be one chromosome or multiple regions, specific genes or a list of specific genes, as we've shown previously when filtering for BRCA1 variants. Or we can filter by some variant type. We can also filter by population frequency in reference populations, for example, the GNOMAD genomes or exomes, or in any other study, 1000 genomes. We can filter by phenotype, for example, using gene ontology terms or human phenotype ontology accession numbers. We can filter using deleteriousness or conservation scores, using tools such as SIFT, Polyphen, CAD, PhyloP, FastCons, and GERP, with a range of cutoffs. Or by protein consequence type. Here you can select pre-specified groups of consequence types, such as all loss-of-function variants, or all protein-altering variants. Or you can individually select the protein consequence types of interest. To apply any filters of interest, make sure to click the search button to apply them to your table. To remove any unwanted searches, just deselect from the filter bar. Finally, we can also filter using clinical databases, such as ClinVar and Cosmic. By a gene's role in cancer, using a free full-text search, or by disease panels. Pre-made disease panels, such as those from Genomics England's panel app, can be loaded, like these here. But it is also possible to create your own using the Clinical Analysis app New Disease Panel tool, as shown in the interpretation review video, which can be accessed through the Clinical Analysis app. Disease panels can be created from lists of genes, variants, genomic regions, or a mixture of all of these, and just need to be selected here. You can type in. Filtered variant tables can also be exported easily. Tables of up to 1,000 variants can be exported in TSV or JSON format using the Export button Download Table option. For example, here, I want to download all BRCA1 loss of function variants. So I go to the Export button, select TSV in the Download Table tab, and click Download. If you want to download a table of more than 1,000 variants, maybe all loss of function misvariant and missense variants present in this study, which is nearly 1 million, use the Export button Export Query tab. Here we can submit a job to download all variants in VCF, Ensembl VEP, or JSON format. The job ID is listed down here and can be monitored via the job monitor, where once a job has been run, it's a variant and export, we can access the output down here in output files.